my name is Catalina Barboza. I'm a master's student at the University of Calgary under the supervision of Dr. Faisal Karim. Today I'm going to talk about my thesis project, which is molecular characterization and vaccine efficacy studies of infectious laryngotracheitis virus in chickens raised in Alberta. Infectious laryngotracheitis virus, or ILTV, causes an upper respiratory disease mainly on chickens. It occurs frequently in intensive populated production areas and causes a reduction in egg production and induces mortality. Birds of all ages are susceptible to disease, and there's been studies that have shown that it can be visible as early as 3 weeks of age. Poor biosecurity is the predominant factor in initiating outbreaks. Besides biosecurity measures, to control the disease, we have vaccines. There are two types of commercially available vaccines. One is the live attenuated vaccines, and how the attenuation is done through these vaccines is from a series of passages in embryonated eggs or tissue culture that then give rise to the chicken embryo origin or CEO vaccine or the tissue culture origin or TCO vaccine. These are the most effective type of vaccines available. However, they have some setbacks. One of them is because the attenuation is not complete, they have residual virulence, which means they're able to be transmitted from bird to bird and even revert back to their virulent form. They are the major cause of outbreaks throughout the globe, and they can also give rise to more virulent strains. On the other hand, we have recombinant viral vector vaccines, which are a much more safer option. However, they are more expensive, they do not reduce viral shedding, and they are less effective than the live attenuated vaccines. ILTV infection in Canada. So what we know about ILTV in Canada, it is that it's endemic in backyard flocks here in Alberta and also in Ontario. The most recent data regarding the nature of the circulating strains in Canada dates back from 2006 in a study done in Ontario, where they found that the main cause of ILTV outbreaks was related to live attenuated vaccines and in a second degree wild-type strains. There's reports of sporadic outbreaks throughout the country in commercial poultry, and vaccine practices vary from province to province. Here in Alberta, we do not vaccinate commercial flocks, and a recent study showed that only 13% of the backyard flock owners vaccinate their birds against the disease. So for the first objective of my research, I wanted to molecular characterize ILTV isolates obtained from ILT outbreaks in Alberta and British Columbia from the year 2009 to 2018. So for our materials and methods, we obtained 46 samples from Alberta and 9 from British Columbia. All of these samples came from positive ILTV outbreaks. We then proceeded to do DNA extraction and then did a PCR for open reading frame A and B partial gene that yielded an amplicon of 1,751 base pairs. However, we ran into some issues here as some of our samples were not yielding positive for this PCR. So we went back and assessed that perhaps some of these samples had a low viral load. So to solve this, we did some propagation in chicken embryo liver cells and embryonated eggs. This helped us have more positive samples for the PCR. After the PCR, we proceeded to do amplicon purification and sent for Sanger sequencing. However, we also ran into some issues here as some of our sequences were giving us results as short reads or loss of signal. So what we thought then to solve this was to design a second pair of primers that targeted a region within this first amplicon. So at the end, we have like different pieces to fill in a puzzle. So this let us have some more samples, and then we proceeded to do some phylogenetic analysis. For our results, we were able to sequence 27 of the Alberta samples. All of them came from backyard chicken, only one had a history of vaccination, and the ages ranged from 1.5 to 24 months. For British Columbia, we were able to sequence 5 of the samples, all for commercial flocks. Only one had a history of vaccination, and the ages ranged from 38 days to 10 months. So for our conclusion, we were able to construct this phylogenetic tree, and our major findings were that outbreaks in backyard poultry in Alberta are mainly strains related to live attenuated vaccines, specifically CEO vaccines, and in a second degree wild-type strains. This is interesting finding since the Ministry of Alberta Agriculture and Forestry recommends the use of TCO vaccines since the year 2009. But we think that one of the possibilities is that the CEO vaccines used previous to this period may have been circulated in backyard flocks since ILTV is a lifelong infection and the virus might have been carried to present period due to bird-to-bird passages. A second option will be that neighboring countries that do use heavily CEO vaccines can act as a transmission source to our province. We also observed that in British Columbia, all strains were related to live attenuated vaccines, specifically CEO. So we see, just as in Ontario and the world in general, CEO vaccines are heavily related to outbreaks of ILTV. For our second objective, we want to determine if currently available recombinant viral vector vaccine protects chickens from ILTV induced by a wild-type Canadian strain, Alberta 63. Some background on this strain is that it classifies as a wild-type strain, or genotype 6. It was collected on September 2017 from birds aged 1.5 months from a backyard flock, and they had no previous history of vaccination. For our materials and methods, we were using one-day-old white Leghorn SPF chickens. We were also using the Inovax ILTV vaccine, which is made with herpes virus of Turkey vector. So for our experimental design, we had 4 different groups of one-day-old chicks. We vaccinated 2 of these groups with the commercial vaccine. Then when the birds turned 3 weeks of age, we infected one of the vaccinated and one of the non-vaccinated groups with the Alberta 63 strain. We continued to observe the birds for 14 days for clinical signs. We also took weight at different time points. Then to assess viral shedding, we quantified ILTV genome load through qPCR in oropharyngeal, cloacal swabs, and feather tips that we took at different time points. To assess the immune response, we took serum sample for ELISA, and we took whole blood samples to quantify a specific subset of white blood cells that are important in the clearance of the disease. Results. One of our first findings was that the vaccine failed to mitigate clinical signs at the peak of the clinical disease. So here in this graph, you can observe that in green, we have the vaccinated infected group, and in orange, we have the non-vaccinated infected group. So we see that the vaccinated group, although it has a slower onset of clinical signs early on, when it comes to the peak of the disease, there's no difference between the groups. The vaccine also failed to reduce shedding through feather tips. So we see this because at 7 and 10 dpi, we didn't see any difference between the infected groups. What the vaccine does do is a help to maintain body weight gain. As we see that throughout the experiment, and even at the end of the experiment, the group with the lower body weight gain was the non-vaccinated infected group. The vaccine also achieves to reduce viral shedding through the oropharyngeal route. So as we see here at 3, 7, and 10 dpi, there's a significant decrease of viral presence in the oropharyngeal swabs in the vaccinated group. Finally, when we were quantifying the specific subset of white blood cells that are important in the clearance of the disease, we saw that there was an increase of this specific type of cell at 5 dpi when compared to the non-vaccinated group. This may explain why at early stages of the disease, the vaccinated group has a slower onset of clinical signs. As a conclusion, we found that the recombinant viral vector vaccine, Inovax, provides a partial protection against the ILTV genotype 6 wild-type Canadian strain. Summary. So for the first part of our work, we did some molecular characterization. For this, we had some positive ILTV isolates. That way, we proceeded to do DNA extraction and did PCR of the open reading frame A and B, and then sent for Sanger sequencing. For our results, we found that in Alberta, the majority of samples classified as CEO revertants and in a second-degree wild-type. For the results in British Columbia, we identified both CEO vaccine and CEO revertant strains. For the second part of this work, we did some vaccine efficacy studies. For this, we vaccinated birds at 1 day of age and then challenged them at 3 weeks of age, and then did some sampling to test for the vaccine efficacy. For our results, we found that recombinant viral vector vaccines confers a partial protection against Canadian wild-type strains. So ILTV in Alberta, we know that it's endemic in backyard flocks and that commercial flocks are not routinely vaccinated. This makes backyard flocks a constant source of transmission for the commercial flocks. So overall, for our results, we found that this work adds to the conversation of the need to develop better vaccines that are as effective as the live attenuated vaccines, but are safer and accessible as well for the backyard flock owners. Finally, I would like to acknowledge my supervisor and supervisor committee, my lab members, the virology group at the University of Calgary, and all the people involved that make this research possible, as well as my funding agencies. Additionally, I would like to thank the people who organized this forum and gave us the opportunity to present our data. Finally, I'm happy to answer any questions that you have about my research.
Thank you very much for your presentation, Mrs. Barboza. This is very interesting. I have a question regarding ILT in backyard flocks. You've mentioned that the vaccine is efficacious in partially protecting against the wild type strains that you've isolated in both Alberta and BC. I was wondering, how is the vaccine administered in commercial flocks?
So this vaccine is administered either at embryo day 18 or in ovo or at day 1 subcutaneously.
Okay, and given that the commercial birds are have a life expectancy, if I can say, of a year to well, approximately a year, are you aware of any studies regarding the protection duration of the vaccine? Because backyard flies have a longer life expectancy than the commercial birds.
Yeah, actually, there's been studies looking into like long-term protection, and they saw that at 35 weeks, there's still some protection, but then at 74 weeks, there's no protection with this particular vaccine. So they're talking more of an approach of doing combination vaccines. So having like, the first dose being this recombinant vaccine, and then later on doing a booster with the live attenuated vaccines.
Okay, and with a live attenuated vaccine, would you suggest then a tissue culture vaccine rather than CEO for those backyard flocks?
So yeah, like here in Alberta, that's the guideline. Like that's what the minister suggested, the use of TCO vaccines. However, misuse of TCO vaccines can also lead to the same issues, as we are also seeing that TCO vaccines, when they're used in a higher dosage, they have given rise also to more virulent strains as well. So it's more about education and also, yeah, when you have to choose between either two, it's better TCO, but also need to have that in mind.
Yes, and then educate the backyard flock owners about biosecurity, I guess.
As well, yeah, that's also very important.
Yes. Well, thank you very much for your presentation. Oh, there's a question from the audience. It has to AB-S63 is a CO revertant or a wild type virus?
It's a wild type virus.